I am going to try my best to give you a landscape or overview of our strategic plan. So we can go ahead and, he's pulling up my slides because, again, I just pulled it off my computer. I hope you guys have been enjoying your time. I know it's very overwhelming. But here we go. We're doing the research roadmap update. And I think we all know this was discovered in 2016, and this is where we started in research with a single publication. And I like to do a little timeline just so people can get a better idea of how far we've come. I'm not going to list everything here, but the foundation was created in 2018. Some of the significant milestones were Jennifer received in 2021 a $50,000 grant from Chan Zuckerberg as a pilot grant, and she did use that money to hire me. And we have really made a lot of progress since then. So I was hired in 2023, as you can see from this timeline. At that time, there was a pretty decent amount of publications, 28, and we had 215 individuals in our registry. We've really grown. We, in 2024, received the largest grant we have to date, $800,000 from Chan Zuckerberg, and it's going to be over the next 5 years. And then this year, we're having this massive conference, which, by the way, started out as a third of its size and has exponentially grown. We now have received 3 grants at the foundation, and we're at 43 publications and counting, and 328 individuals and counting. So you might have heard us talk about a lot of tools in the discussions lately. We've really tried to build up our research toolbox such that if a researcher has an idea or a question they want to test, that they don't have to go make something from scratch. That's not saying they never will have to, but we've built this nice toolbox with 4 different mouse models. We now have 2 different natural history studies ongoing. We've got flies. We have induced pluripotent stem cell lines, which are directly taken from patient samples. And then we have some other cell lines called fibroblasts that we can use to study different things about OCNDS. I know you all have seen many different versions of this figure, but I've added here on each segment of the protein the number of people in our registry that fall into these different regions. The white regions between the colored ones we just call interdomain segments because they don't really have another classification as of yet. And so that number there is in our legend with the colors. So there's 62 people in the interdomain segment. The most patients we see are in what's called the P+1 loop. That's that dark blue, and that's where that K198R mutation is. And then we do have quite a significant number of patients in that pink region, which is the glycine-rich binding loop. And I really want to thank Elena and Maheen for making this beautiful figure that's color-coded to the regions. So you can try to gain an understanding of how complex this protein is. And when I say loop, it literally is a loop. I've intentionally left this blank. Some of you, if you've seen my previous presentations, have seen this type of roadmap. And it's a generic roadmap. Any type of rare disease foundation can use something like this, but it's really the basic structure that we need to move forward. So we're starting with natural history, which you've heard a lot about lately, so we can measure the progression over time. And one of the key unanswered questions that's come up, not only for families, but also for researchers and clinicians, is we don't have a lot of adult patients to learn from. We actually don't know how it progresses. And that's why natural history is so important. We can then combine that with different disease models as well as basic science. So that's like the nitty-gritty things that you're hearing about with the phosphorylation of proteins and where is the protein in the cell and what does it do when we mutate it downstream. But we need all of those 3 things that then take it into what we call preclinical, so prior to going into a clinic, or translational research. We hope in the near future that we will then lead into phase 1 to human studies, to move on to longer phases, and then science doesn't sleep, research doesn't stop. There's always going to be long-term monitoring. So I hope I can fill in some of these gaps for you along the way. One of the key things that came up in our researcher talk is called genotype-phenotype comparisons. I know that's also a mouthful. What we mean by that is, does the place where the mutation is in that gene affect how that patient presents? So what symptoms do they have, how severe they are, what they will be when they progress. In 2024, this paper came out, And one of the key findings is that people who have what we call null variants, and that's a variant that does not result in a gene product. It's not every variant, but some of the nonsense, frame shift, deletions, if you've seen that on your genetic report, that could be one of these. They exhibit maybe a reduced amount of symptoms in specific domains like language, the facial features, intellectual disability, and then what they're calling an overall milder phenotype. So I looked at this and I said, okay, well, most people with OCNDS have a missense mutation or a one-letter change. What about those? What can we learn about that? So in collaboration with two amazing interns, Elena, who's here, Maheen unfortunately couldn't be here, but I do just want to put a plug that he is working with us from Pakistan. It is a very big time difference, so he was spending his 8 to 10 p.m. hours with us, or extremely early, all the way from Pakistan. I think that's pretty amazing. And we did this virtually for a year and a half. And one of the things that came out of this, which some of you have seen this slide earlier in the conference, this is data on 48 people in Simon's Searchlight, and it's only people with missense variants that had sufficient data, and they were classified as either pathogenic, disease-causing, or likely pathogenic. And what I hope you can appreciate, when we're looking at the up and down to the vertical axis here, that's the total number of patients out of that 48 that has the symptom that you're looking at on the horizontal axis. So that first bar in black is total number of patients is 48. As you can see, 48 out of 48, 100% had a speech-language delay or a disorder. In the literature previously, it was only estimated to be 34% of patients. So this is a huge difference. That's not saying we won't be, disproved in the future as we look at a larger cohort. But as we've heard today in our discussion, speech and language is a huge issue. We're also seeing common features like neurological, global developmental delay, gastrointestinal. Pick your favorite one, you're going to see it. But this is what we found so far, and that paper is out and available to read. I'm also going to point out, I don't even think Elena knows this, I did this at 1:30 in the morning last night. This paper now has over 1,300 views and 140 downloads. It just came out. Amazing. So if you're one of the ones, thank you. To further look at genotype, phenotype, this is also from the paper, but I do think it's worthwhile showing you guys. We tried to look at the total number of symptoms as a proxy for disease burden. As you've heard us talk about today, we don't have a clinical severity score. We are now looking at each region of the gene, and the intradomains are the white, so those are grouped together, and the number of symptoms that they have. And what we first noticed was in the pink column and the red column, which is broken out into 3 separate loops, we see a lot more dots. Not only number of patients, but they're going up higher in the plot, meaning they had more symptoms. So we decided to then group people who had mutations in what we were calling the loop regions versus non-loop. And what we found was that people who had loop variants were often diagnosed at a younger age. They tended to have a higher trend of symptom number. And they presented with a statistically significantly higher proportion of people reporting low muscle tone. So there are some genotype-phenotype differences. Now, these surveys are very restricted. They're only asking specific questions. So we then wanted to ask, what symptoms impacts, what does it mean most to families? So Grace is actually going to be following me. She's going to be talking about her disease concept model, which is a project with Combined Brain to better understand the lived experience. And as a foundation, we've chosen projects to address the most impactful symptoms. One is the speech-language function, which we're about to hear about. We've heard about 2 different sleep projects, one with Dr. Alada and Jack Parent, looking at biomarkers, as well as Vishnu with the flies. And then to put it all together, right, we've heard about all these disparate projects and all over the world. To put it together, Dr. Dennis Law is helping us to create a knowledge base, which will be a resource not only for the researchers, but also the families. And I'm going to show you a snippet of that in just a minute. And if you've participated on site in the neurological assessments, this is really important because we're using wearables now to learn more about the neurological phenotypes. This is just a screenshot from a portal from a different gene, not to confuse you, but to give you an example of what we're going to put together, a history of the research, educational resources for families, specific variant analysis, and then we're going to put all raw data that were given by researchers into one spot, so it's going to be a one-stop shop for everybody. There are additional projects that's going to guide our therapeutic approach. I'm putting them all here. You've heard about most of them here: 2 drug repurposing projects, mouse model characterization with the GI project, the pluripotent stem cell characterization, cilia perturbations with Dr. Lukiel, and then the caregiver interviews to assess milestones with Dennis Law's team. So what is that leading us to? We have new suggestions from this meeting. I'm emphasis on not limited to these, but finding older patients to learn about the progression, determining the disease mechanism for each variant, which for families, what I mean is a biomarker or a readout for disease presentation that's likely going to differ. Initiating longitudinal sample collection now so that we can build a robust biobank for future projects. And then we've also determined that we're going to establish some smaller working groups of researchers and clinicians to focus on common themes, so sleep disruption, drug repurposing, outcome measures. So when we put most of this all together, we ended up filling in this roadmap. You've heard a lot about different parts of it, but I just wanted to lay it out here. These slides will be available to you. We're running short on time because things happen. So I'm going to skip over running through it, but I'll give this to you guys. The only thing I do want to mention is we are really trying to get an ICD-10 code, which are those codes that your doctors plug in when they give you an autism diagnosis or something else, so that we learn more about people as they're being diagnosed. So I put this here as a brief reflection. I am tattooed, and I have a bee tattoo on my wrist, and I often get asked why I have a bee tattoo. And one of the people who first asked me about this is Jackie Lopez, when we met at an epilepsy conference that she came to volunteer at and help me out with a booth. I chose it. I actually flew to Phoenix for a special fine line tattoo artist. I ended up meeting with Matt from TGen there. We had a great discussion. And the next day I got tattooed, and I chose a bee to remind me of something really important. Bees are really small, but they're incredibly hardworking, and they're vital to our world. A single bee can visit up to 1,500 flowers per day. Collectively, they pollinate up to 75% of all flowering plants. So their tiny efforts lead to a huge, thriving ecosystem. In the rare disease space, our individual actions, even small ones, really can add up to build up in the same way. Our daily contributions can create change that lasts. And I will say, I will never in my life think that I would be an influencer, but the picture you're seeing up here is Jackie and Maddie that now have matching bee tattoos with me. So thank you. And with that, a huge thank you for all of you being here. A huge thank you to the AV team, Combined Brain, all of our researchers on site, our interns, board of directors, parent advisory board, scientific advisory board, families participating in research, and last but certainly not least, the best boss I've ever had, Jennifer. Thank you.